So far we have seen that B cell receptor, also we have seen T cell receptor, but I told a couple of times at least that B cell receptor can recognize antigen directly. Okay, so B cell receptor can recognize antigen directly. So if there is an antigen, B cell receptor can directly attach to them. But I already told that T cell receptor cannot recognize the antigen directly. For that what we need? For that we need, it should be presented by MHC, major histocompatibility complex. So this lecture, now I am going to discuss about antigen recognition by T cell. Actually we are going to talk about, mostly in the initial part, the main theme is antigen recognition by T cell, but initially we are going to discuss about major histocompatibility complexes, MHC. Okay. So you already know that T cell receptor recognize antigen in the form of a complex of foreign peptide which is bound to MHC. So if it bounds to MHC, only then it is going to be recognized by T cell. Okay, and if I will show you now one picture which is already I have shown you, that there are two classes of MHC molecules, right? They are very distinct subunit composition, but they are three-dimensional structure, or they are just, if you look suddenly, they are very similar looking. Okay, this was the simplest cartoon that already we have discussed. This is MHC1, this is MHC1, and the other one is MHC2. This is MHC2. I mean, if you see the yellow box or solid part of yellow, it's very, very same, right? And only difference what we can see here, there is one transmembrane domain, and here there are two transmembrane domains. These things already we have discussed in the basic part or the initial part of our lecture. But if you see detail, how this structure or this kind of cartoon came up, it is most of the protein structure, it is came from crystallographic structure. Don't be scared by all this figure, I will explain very slowly. This is one way presentation of the crystal structure of a protein. Okay, there are different domain of different color. This is alpha helix and beta sheet. This is another simple way that we can understand. And this is just the cleft, this region. Okay, this region. Cleft means where the antigen binds. If you see this region, let me take a pen, that will be easier for me. So this region is, if you see, like if you see something, if I show you something like this, okay, this way you can see the outline here. But if you see this, you can see the outline here, only the outline. But how this looks inside, you can see only if it is do like this. So how the inside is, you have to see how flat it is, what is the space. You can see if I show you this view, right? Top view. Okay, this is the side view. This figure is this is the side view, how it looks sidewise, and this is the top view. So this is actually the cleft. This region is the cleft. So it is like a plate or a dish where there are some ridges there. Okay, so something should not fall apart, and there is a space between. So antigen can fit here. I mean, in this region, in this pump top, it can fit the antigen, and this is the boundary. So this region, the peptide binding cleft, is basically the pump. And if you make it simpler version, this is the peptide cleft. Okay, this is the peptide cleft, and this is overall structure. You see again here also the binding site or the peptide cleft is mostly the beta sheet and alpha helix, and the major structure or the framework region, overall structure maintained by mostly the beta sheet. Okay, it is very similar structure of globulin molecule. Okay, so now if I see one by one, so this is the special model, and you see the different domain has different color. I'll see this is again, this is in better, this is the peptide plate. Okay, this is alpha helix, these two are alpha helix, and rest part and the plate portion, where the flat portion of the plate are beta sheet. Okay, this is the same picture what I showed before. So this is the alpha helix, and this the flat portion is the beta sheet, and this is again another alpha helix. Okay, two alpha helix and beta sheet is making the plan. And now if I see how this structure is, you see there are two peptides actually. One peptide has three domains: alpha 1, alpha 2, and alpha 3. 1, 2, and 3. Okay, alpha 1, alpha 2, alpha 3. This is one polypeptide chain which has 1, 2, and 3 domains. And that domain, that 1, 2, and 3, along with that, this is the transmembrane domain which is integrated here along with the membrane. Okay, the transmembrane domain was just fixed in the membrane. So alpha 1, alpha 2, this is the outer. So this is cytosol, and it's inside the cell, and that is outside the cell. So this is outside the cell. Okay, so it is exposed to the outside of the cell. This is cell membrane. Okay, this is the membrane. So alpha 1, alpha 2, and alpha 3 is one polypeptide chain, and another protein, it is just like if this something is like there, and if you need a support, okay, so that it should not fall apart, you need a support kind of thing. So this support is the another peptide named beta microglobulin. Okay, so these three domain is there, and beta microglobulin is there. It is not covalently linked. Okay, it is just a protein-protein interaction. So something is like that, and it is giving a support. Clear? Now I am going to go to MHC. This is MHC1. So MHC1 constituted of two peptides, alpha 1, alpha 2, alpha 3, one peptide, and beta microglobulin gene, or beta microglobulin, which is giving the support or making the MHC complete. MHC1 a complete structure. Same way, if you see the MHC2, you see suddenly if I don't show you the previous picture and we just drop this part, okay, this region, if we don't consider for the time being, then what will happen? You see, it's very similar. You can remember what you have seen before, right? This same 4 domain, same 4 color code. Their peptide binding plate is exactly similar looking. They have a beta sheet in the flat portion, and this peptide binding plate again looks like same. Okay, this beta sheet is here, alpha helix are here. Only thing, if I see this cartoon, then I can see that no, this is not really exactly same as MHC1. Okay, crystal structure is very, very important to know all these details. But crystal structure determination is one of the toughest thing. Even today there are so much advance in crystallography and data structure analysis, but even today it's very, very tough. Okay, first hurdle is protein purification. You need a very pure protein. Okay, then you need to find a condition where it will be crystallized. Then you have to, that crystal should diffract properly. Okay, many times the crystallographer got nice looking crystals, but the diffraction is not, the resolution of diffraction is very, very bad, so you cannot get the proper structure. Now a different technology, cryo-EM techniques for protein structure is here, so now life is much more easier than crystallography and data structure, X-ray diffraction. But even today, the problem of this kind of structure, like MHC, structure of MHC, is very, very tough. Why? I'm telling all these things just to give you an idea, like this thing is okay, there is no point, why people are doing this, this is very simple. No, it's not that simple. because first point you have to remember, it is there in my next or some slide later, the MHC molecules are very unstable until unless they are bound to peptide. This structure is not very stable if there is no peptide attached to it. Second problem, that MHC molecule is a membrane bound protein. All membrane bound protein has a strong hydrophobic region, and to make protein, or you need to dissolve the protein to get crystal, and hydrophobic proteins are not dissolved easily in water, so you have to find a solution. And third point, and which is very important, then isolation of membrane protein is really tough. Okay, there is no one buffer or one system that all membrane protein will come and will be in the solution. And because our cell membrane is not very straightforward, right? There are a lot of lipids are there, fatty acids are there, and there are a lot of proteins are there, some are embedded. So that lipid bilayer you have to dissolve first. And to do that, and there are some certain region, I'm not going that much detail, but to dissolve this lipid and to get that protein out from that lipid, you need a very hard solution. Okay, it is not that very quick, mild detergent can do anything. Many times, to dissolve the protein, to rupture the membrane, to dissolve the protein, your buffer should be very tough. And in that tough condition, I should say more scientifically, it is denatured the protein. And if the protein denatured, then it will lose its structure, you cannot determine it. So any membrane protein structure determination is very tough. In this case, it is even more tough because MHC molecules are very unstable without peptide. Okay, and as soon as you give a strong denaturing solution or detergent or something, that peptide will fall apart because this is not a covalent bond. As soon as it fall apart, MHC molecule will be unstable. So these structure, what you will see in this figure, okay, very simple and easy. It's not that easy. Okay, so this is just to give an idea. And after doing all these things, even today, not many MHC molecule structure has been determined. But so far, some scientists spend too much effort and sincerity. That's how we can tell or we know something about MHC. So from that crystal structure, whatever we are going to tell, that is not going to take long time. I can tell you it's very simple because I already told you that alpha 1, alpha 2 and alpha 3 is a major part of the MHC1, and support or the other component is the beta microglobulin, which constitute the MHC1. If you see MHC2, then it is two polypeptide chain. One is alpha, this one is alpha, and another one is beta. Okay, so both of them, alpha also has two domains and beta chain also has two domains. Both of them independently they have a transmembrane domain. So alpha 1 is impregnated in cytoplasm, alpha chain also they are have a transmembrane domain which is fixed in plasma membrane, and beta chain also has a transmembrane domain which is fixed in plasma membrane. So they come together and form this cleft. Okay, so this cleft is contributed by two different chains, alpha and beta. So now I will tell some difference slowly so that you can remember what is what and how their function is different. So this is again the same thing what we discussed in bigger format. So if you go there, this is the same thing, same crystal structure, alpha helix and beta sheet. This is again peptide binding cleft. This is for MHC2, and this is that alpha 1, alpha 2 which is contributed, and this is the peptide binding cleft of MHC2. It has two transmembrane domains. So now I'll see this. See, this one is MHC1 and this one is 2, 1, 2, 1, 2. just a quick look, you will find very similar, not much difference at all, right? But they have number of difference which make their function different, and also their location is they're located inside the cell in different places. Their function is different. I don't remember, even if I tell, I am repeating again. MHC1 is activating cytotoxic T cell. MHC1 is activating cytotoxic T cell. Yes, I told you, because if you remember the virus protein, how it is going outside, because virus protein is presented by MHC1. So any internal protein or endogenous protein is presented by MHC1. Any external protein or protein coming from outside by phagocytosis or pinocytosis, that is presented by MHC2. So two complete different pathways. So their role is different, even they look similar. Okay, so what is the difference? Definitely there is a difference in 1 and 2. I am not going. So first difference, MHC1 has one transmembrane domain. So transmembrane domain, with respect to transmembrane domain, MHC1 has 1, MHC2 has 2 transmembrane domains. Clear? Second difference, in MHC1, peptide binding cleft is constituted by one protein only. It is a continuation of one protein, even they are very similar looking. Okay, even they are very similar looking, this one and this one very similar, but here alpha 1 and alpha 2, this is MHC1 and this is MHC2. Alpha 1 and alpha 2 are contributing this MHC1 peptide binding cleft. And in case of MHC2, peptide binding cleft is contributed one by alpha 1, another by beta 1. So how it is different? And why it is? They are exactly same looking, same space? No, there is a difference. How? Because even if you have a very big hand, there is a limit in one hand. Okay? There is a limit in one hand. So in one hand, if this one hand, why I am telling one hand? Because this is contributed, suppose the finger part is alpha 1 and the palm part is alpha 2. Even the fingers are very big and palm is also very big. You have a limit how big thing you can hold. I am talking about the peptide here. So in one continuous thing, if this is the alpha 1 chain, if you consider this hand is my alpha segment, so this finger is alpha 1, palm is alpha 2, and this arm is alpha 3. If you like that, then you have a limit. You cannot hold any substance, or rather you can hold a substance up to certain level. If it is too big, you can hold, right? If your hand is big, you can hold a basketball or a football very easily. But if I double the size of basketball, one hand may not be enough, right? But if this thing is contributed by two hands, even the size is same, assume that my two hands are half size, so that it covers this size only. Or you think a kid whose hand size, total hand size is half of my hand, even after that, two hands is good enough to hold bigger, because we have a flexibility. I can hold things like this. One hand I have a limit, but two hands I can hold much bigger thing than a single hand. So here is the difference of MHC1 and MHC2 also, because in one peptide cannot hold bigger peptide. One alpha chain cannot hold a bigger piece of peptide, but MHC2 can hold bigger peptide. It can accommodate much bigger peptide in this peptide binding cleft. So this one chain is not, two chain is not just simple. So it has a variation to, because whatever the peptide they are going to bind or bind in the cleft, it is going to be displayed, which will be recognized by T cell, which will be recognized by T cell. That we have discussed before. And in the peptide binding cleft also, there is slight difference. Let's go for for the next slide. So let's see what is there. You see here in this is MHC1 and this is MHC2. How you will figure it out? Because you see same color code. So alpha 1, alpha, both are alpha, so all are yellow here, and here two different chain, two different colors. One is yellow, another is green, right? So this is alpha 2. But if you see this peptide size, if you see the peptide, the red one is the peptide. This peptide size, this is real. I mean, it is not just a drawn picture. It is a real crystal. This structure is determined from a real crystal where MHC along with the peptide was crystallized and structure was determined. You see the length of the peptide here is much less than MHC2. So MHC2 can hold bigger peptide. I'll come what is the number and how big it is. So peptides are stably bound to MHC molecule and also serve to stabilize the MHC molecule on the cell surface, that I just mentioned few minutes back. MHC molecule are only stable on the cell surface if peptide is attached to it. So only free MHC you will never see any free MHC on the cell surface. It is always peptide bound form. Okay. Now the number. So MHC class 1 molecule binds shorter peptide in comparison to MHC2 molecule. So MHC class 1 molecule, how short it is? It can bind 8 to 10 amino acids. It can bind 8 to 10 amino acids. In few books, even in same GNOA book that I am following, if you go to older version, you may find in 9 to 11. Okay, 9 to 11 amino acids. So it is not exactly, because there is no fixed rule. It is average 8 to 10, maybe plus minus 1 or 2. Okay, so 8 to 10 amino acid length peptide MHC1 can bind. In case of MHC2, it can bind approximately 13 to 20. So the peptide length is 13 to 20. If you remember our discussion in the early lecture, what we have seen, that big protein chopped into pieces, a small piece goes to peptide. So that small piece size for MHC2 is 13 to 20 amino acid length, and in case of MHC1 it is 8 to 10. This is another difference. So one can hold bigger peptide. How big? You know now. And another MHC1 is smaller. How small? You know now. So if you see this is MHC1, this is MHC1 with the peptide. Okay, if you see this is 8 to 10 amino acid peptide, and this peptide, this is the peptide binding cleft. Okay, this is the alpha helix, this is the beta sheet in between, and if you see carefully, what you can see is, you can see the attachment, because it is again a protein-protein interaction, right? Because MHC is a protein and peptide is also a part of the protein, so it is non-covalent interaction. So it is again a protein-protein interaction which will follow same like hydrophobic, electrostatic, van der Waals forces, kind of non-covalent hydrogen bonding, non-covalent bond, interaction. So these interactions, either one or all, possible combination of this non-covalent interaction, it is happening in the end part mostly. Okay, so middle part of the peptide is not interacting. This is in case of MHC1. And this is the analyzed peptide. So what happened? Different peptides were tried. Is this binding specificity as specific as antibody antigen interaction or T cell receptor peptide interaction? No. They can accommodate smaller size of peptide, but their specificity is not that strong or that specific like antibody antigen interaction. Here, suppose this is 8 nucleotide peptide, And what scientists did, they tried to feed variety of peptide to see which is, for a particular MHC definitely, which is binding better than rest or which is the best binding. What they found that in this, for example, in this case, the C terminal should be always leucine, okay, and this one is, you see, the organic, tyrosine or phenylalanine. Rest may be anything. So, position of this particular peptide is very important to interact. Similarly, this is another. This is the example of another set of peptide, which is again this the C terminal peptide, and this here it is though conserved, all should be tyrosine, but rest is not that important, and this is not true for antibody. Antibody cannot be that generous. Okay, why? Because we have so many antibody in our system, variety of antibody, which can accommodate any kind of antigen almost, but we don't have that many MHC molecules. We have a limited number of MHC. Okay, say for example 200 per individual. Okay, so these 200 average cannot present only 200 types. So what is happening? One MHC can display variety of peptide. They only specific only 2 or 3 regions. In this case, MHC1 only 2 specific sites or location. If it is okay, rest the MHC does not care. So one MHC can display variety of peptide where sequences are different only if this thing is maintained. What is this thing? This say the C terminal case, in this case of all leucine, in the N terminal all should be tyrosine, and here valine, isoleucine, leucine. Okay, but if you see, this is all organic, this is all hydrophobic, right? So this all are hydrophobic. So there is a pattern. It is not like any amino acid. So why this is? Similarly, if we go for MHC2, MHC2 binding, not only it is bigger, you see it binds almost throughout the cleft. End is definitely it is interacting in the end, but it is interacting throughout the cleft. Again a difference. MHC1 binds mostly at the end, but MHC2 binds with the peptide throughout its cleft. All this non-covalent interaction happening throughout its cleft, and this is another example the same way. This is a variety of peptide generated and tested with a particular MHC. What is their flexibility? All the green one, they are rigid. Okay, not exactly 100% conserved, but you can say there is a pattern definitely. But if you see like this, all this most of except one peptide, this one except this one, all have aspartic acid here, except one is glutamic acid, right? Same way you will see the pattern. So they can accommodate not, variety, their length is also varying. Okay, they accommodate bigger and more flexible. Their interaction pattern is different. So these are the different. If there is a question comes in exam, like what is the difference between MHC1, or compare MHC1 and MHC2? Okay, structure and function wise. Structure definitely you can tell, and this is the function. Function is one is presenting to cytotoxic T cell, another is presenting to helper cells. Their peptide length is different, their interaction pattern is different, their peptide tape is made by one peptide and two peptide, two transmembrane domain, all are coming to the comparison between MHC1 and MHC2. Their locations are different, that we'll see later, maybe in the next lecture or next to next lecture.